Hello there and welcome to the deep dive. It's great to have you with us today. We're about to embark on, well, a really fascinating journey right into the architecture of plants. Our mission today is to really get to grips with the anatomy of flowering plants. And this isn't just another chapter you flip through, it's genuinely a foundational cornerstone, absolutely crucial for your class 11th and 12th board exams, and maybe especially for competitive exams like NEET. We're going to peel back the layers quite literally and discover how these incredible organisms are put together, from the inside out.
Hello. Yes, I'm definitely ready to dive in. Anatomy of flowering plants. It's always highlighted as a high weightage topic.
Right.
So yeah, I'm looking forward to understanding all the core concepts and the specific details we really need for the exams.
Excellent. That's precisely our goal today. You see, when we talk about plant anatomy, we're essentially delving into the study of their internal structure. Just think about the huge variety you see on the outside, right? Like the towering trees, delicate flowers, sprawling vines. Well, there's an equally intricate and diverse world hidden within them. And even within the same group, say angiosperms, if you compare a monocotyledonous plant to a dicotyledonous one, their external look might seem similar sometimes, but their internal blueprints, they often reveal these fascinating, really distinct variations. Understanding these variations, that's where the real knowledge lies. And honestly, that's where the crucial exam marks are often found. Okay, so at its most fundamental level, a plant, really like any living organism, starts with cells. These aren't just, random cells floating about. No, they are meticulously organized. They form specialized groups we call tissues. And then these tissues, well, they come together in highly coordinated ways to form the various organs of the plant. Think roots, stems, leaves. What's truly captivating and what you need to grasp is that each of these organs has its own unique internal structure, and it's perfectly adapted to perform its specific set of functions. It's like an engineering marvel, really, a highly efficient blueprint for life.
Okay, that makes sense. But sir, if we can see a lot about a plant just from its external appearance, its morphology, why is it so, critically important for us to spend so much time studying these internal structures, the anatomy? What's the real benefit, especially thinking about exams?
That is an absolutely brilliant question, really sharp, and it cuts right to the heart of why this topic is so vital, especially when you're gearing up for competitive exams like NEET. Look, while the external appearance gives us clues, definitely, it's the internal anatomy that truly unlocks the how and why behind a plant's survival, how it succeeds in its specific environment. For instance, picture a desert plant. Right. From the outside, you might see thick leaves, maybe a fleshy stem, okay? But it's only by studying its internal anatomy, the modified tissues for water storage, maybe those sunken stomata, the thick cuticle, that you truly understand how it manages to thrive with so little water. These aren't just abstract facts to memorize, they are real-world adaptations. And for your exams, this conceptual understanding, this ability to connect structure directly to function and adaptation, that's absolutely crucial for tackling those challenging application-based questions. Those are the questions that really differentiate the top performers, and this deep dive today will equip you to handle them. All right, then. So with that crucial foundation laid, let's now try to organize our understanding. Let's look at how these structures are actually classified. Tissues within a plant, they are far from uniform. Their characteristics, their roles, they vary significantly depending on their location. And of course, their specific structure and function are intimately linked to where they're situated within the plant body. It all fits together. So based on these aspects, their specific form and their placement plant tissues are broadly categorized into three major tissue systems. Let's take this step by step. We'll break down each one carefully. These three fundamental systems are the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. Okay, let's begin our in-depth exploration with the first system, the epidermal tissue system. This system essentially acts as the plant's outermost protective shield. Think of it as its first line of defense against the external world. It's like the plant's skin, really, safeguarding it from various environmental stresses, pathogens, and maybe most importantly, excessive water loss. This system is actually quite a sophisticated complex. It comprises three key components. First, the epidermal cells themselves, which form the main covering. Second, specialized structures called stomata, which are absolutely crucial for gas exchange. And third, various epidermal appendages, which include structures like root hairs and things called trichomes. Now let's zoom in on the epidermis itself. It forms the outermost layer of the primary plant body that just means it's developed during the plant's initial stages of growth. Typically, the epidermis is a single-layered structure. It's composed of elongated cells, usually parenchymatous, that are highly compactly arranged. Think of them like perfectly fitted tiles, right? Forming a continuous, unbroken barrier. Inside these epidermal cells, you'll find a relatively small amount of cytoplasm, usually lining the cell wall, and often a large central vacuole. And that vacuole is incredibly important. It helps maintain turgor pressure within the cell, which in turn gives rigidity to the plant body, keeps it firm. Okay, moving outwards from the epidermis, you often find a remarkable feature covering its outer surface, a waxy, often thick layer known as the cuticle. This cuticle serves a vital primary function. It significantly reduces water loss from the plant through transpiration. Remember, transpiration is the process where water vapor escapes from the plant, mainly through its leaves, into the atmosphere. So this waxy layer acts like a natural sealant, a kind of waterproof coating. It helps the plant conserve its precious water. And that's especially critical in, say, arid or hot environments where water is obviously a limiting factor.
That makes perfect sense for parts like leaves and stems, where water conservation is obviously key. But what about roots? Does the cuticle cover them too? It just seems a bit counterintuitive, if roots are meant for absorption, right?
That's an incredibly sharp observation. Really good thinking. And it highlights a crucial detail that's often tested in exams. You're absolutely right. The cuticle is specifically absent in roots. And your intuition. Spot on for the reason. Roots have the fundamental role of absorbing water and minerals from the soil. That's their main job. Now, if they were covered with first, transpiration, which is that controlled loss of water vapor from the plant, and second, gaseous exchange. This involves taking in carbon dioxide, the raw material for photosynthesis, and releasing oxygen, which is the byproduct of that process. Right, let's break down the structure of a stoma slowly, step by step. Pay close attention here because this is a very, very frequently asked diagram-based question. You'll see it in practicals and theory exams. Okay, each stoma isn't just a simple hole. It's a precisely engineered little apparatus. It's composed of two specialized, usually kidney or bean-shaped cells. These are known as guard cells. And they meticulously enclose the central stomatal pore. Now, here's an important diagnostic detail, especially for identifying plant groups. In grasses, which are typical monocots, these guard cells aren't bean-shaped. Instead, they are distinctly dumbbell-shaped, like a little weight dumbbell. This specific shape is a key identifying feature that helps you differentiate monocots very quickly. Okay, now pay close attention to the cell walls of these guard cells because their structure is absolutely key to how they function. The outer walls of the guard cells, the ones positioned away from the stomatal pore, they are relatively thin and quite elastic. But in stark contrast, the inner walls, the ones directly facing towards the stomatal pore, these are highly thickened and much less flexible. This differential thickening is absolutely crucial for their ability to open and close the pore effectively. And here's another unique feature. Unlike most epidermal cells, which typically lack them, guard cells possess chloroplasts. Now, why is this significant? It means they can actually perform photosynthesis, which generates energy, right? This energy, in turn, helps regulate the osmotic potential within the guard cells, controlling their turgor pressure. So when turgor pressure increases when they fill up with water, the guard cells swell and they bow outwards because of that differential wall thickness. That opens the pore. When turgor decreases, they lose water, they become flaccid, limp, and the pore closes. Think of it like this. The stomata are like the plant's tiny adjustable gateways. The two guard cells, they're like diligent security guards. And their internal water pressure, their turgidity, is what dictates whether the stomatal pore swings open or clamps shut. It's an incredibly precise regulatory mechanism, finely tuned to the plant's needs and environmental conditions. Right. Now, sometimes a few epidermal cells that are right next to, surrounding these guard cells, they also become specialized. They change their shape and size. These are called subsidiary cells or sometimes accessory cells. They basically play a supportive role, helping the guard cells in their function. And here's a collective term that you'll definitely need for your exams. The stomatal aperture, which is the pore itself, plus the two guard cells, plus the surrounding subsidiary cells. All of that together forms what is known as the stomatal apparatus. This entire apparatus, aperture, guard cells, subsidiary cells, is a very, very common topic for diagram-based questions. So make sure you can visualize it, draw it, and label it clearly. Okay, finally, within the epidermal tissue system, we have various epidermal appendages. These are essentially specialized outgrowths or hairs on the plant surface. There are two main types you absolutely need to be familiar with. First, root hairs. These are quite remarkable. They are unicellular elongations, just single-celled extensions of the epidermal cells in the roots. Their primary and absolutely vital function is to dramatically increase the surface area available for absorbing water and dissolved minerals from the soil. Honestly, without them, plants would really struggle immensely to take up enough resources. They are critical for the plant's growth and survival. Second, we have trichomes. These are essentially the hairs you find on the shoot system, typically on stems and leaves. Now, unlike root hairs, trichomes are usually multicellular, and they show an astonishing diversity in form. They can be branched or unbranched, soft and delicate, or maybe stiff and spine-like. Some trichomes can even be secretory, producing substances like sticky resins or, stinging compounds, like in stinging nettles. Their main function, particularly on the shoot system, is usually to help prevent excessive water loss due to transpiration. How? Well, they create a kind of humid microclimate right around the stem or leaf surface. This reduces air movement and slows down evaporation. It's effectively like a tiny protective blanket against drowning out, against desiccation. Right, moving on to our second major tissue system. Let's explore the ground tissue system. This system is truly the bulk, or you could say the core of the plant body. It basically encompasses all the tissues that are not the epidermis, which we just covered, and not the vascular bundles, which we'll get to next. It's essentially the filling material, if you like, but it performs really crucial functions. It's not just packing. Okay, the ground tissue system is primarily composed of what we call simple tissues. You've likely encountered these fundamental cell types in earlier chapters, but let's have a quick refresher, maybe a memorable phrase for each. These include parenchyma, collenchyma, and sclerenchyma. Think of it this way. Parenchyma cells are the versatile all-rounders, or the workhorses. They're involved in nearly everything: storage, photosynthesis, secretion, even healing. Collenchyma cells, they're the flexible architects. They provide flexible support, especially to young, growing parts, letting them bend without breaking, very important. And sclerenchyma cells are the rigid engineers. They provide strong mechanical support and rigidity, giving mature, non-growing parts their strength and protection. Think of nutshells or fibers. Now, where do we typically find these parenchymatous cells, which make up a major part of the ground tissue? Well, you commonly find them in specific regions like the cortex, which is the region just beneath the epidermis. Also in the pericycle, an important layer inside the root. The pith, often found right at the center of stems, and the medullary rays, which help with radial transport within stems. These locations are typical for primary stems and roots. Now, a very special and absolutely vital component within the ground tissue system, specifically found in leaves, is the mesophyll. In leaves, the ground tissue is highly specialized. It consists of thin-walled cells that are just densely packed with chloroplasts. This specialized tissue is precisely what we call mesophyll, and this is where the real magic of photosynthesis happens, that incredible process where plants convert sunlight into chemical energy, making their own food. It is the primary site for almost all food production in the entire plant kingdom. Really fundamental.
Okay, so just to clarify then, mesophyll is essentially a highly specialized type of ground tissue, specifically adapted for photosynthesis in leaves, right?
Absolutely. You've grasped that perfectly. It's a prime example of how a general tissue system, like the ground tissue, can undergo remarkable specialization. It modifies itself to perform a crucial vital function like photosynthesis within a specific organ like the leaf. Understanding these specializations and why they matter functionally, that's incredibly important for those exam questions that go beyond just simple definitions. Okay, now let's transition to the third and final major tissue system, the vascular tissue system. As its name strongly suggests, this is the conducting system, the transport system of the plant. Think of it as the plant's internal highway network, efficiently moving essential stuff throughout the entire plant body. This intricate system is made up of what we call complex tissues, the xylem and the phloem. The xylem is primarily responsible for the transport of water and dissolved minerals, usually unidirectional, drawing them up from the roots to all other parts of the plant, like a vital pipeline. The phloem, on the other hand, is responsible for transporting the food, mainly sugars made during photosynthesis in the leaves, to all other parts of the plant. That includes roots for storage and growing regions needing energy. Together, these two complex tissues, xylem and phloem, are organized into distinct units known as vascular bundles. You can visualize these vascular bundles as the plant's essential transport arteries and veins, making sure water, minerals, and food reach every single cell where they're needed. Now, vascular bundles themselves can be further classified, and this is based on a critical structural feature, the presence or absence of a specialized tissue called cambium. This distinction is profoundly important because it dictates whether a plant can grow thicker, increase its girth. First, we have open vascular bundles. These bundles are characterized by the presence of this meristematic tissue cambium located strategically between the phloem and the xylem. Where do we usually find them? They are a hallmark feature of dicotyledonous stems. Now, what's their significance? Well, because the cambium is present, these bundles possess the amazing ability to form secondary xylem and secondary phloem tissues. This whole process leads to an increase in the girth or thickness of the plant, something we call secondary growth. It's like an open road, allowing for future expansion. It's what allows trees to grow massive and strong. In contrast, we have closed vascular bundles. They're named perfectly because they lack cambium completely. There's no cambium between xylem and phloem. And as a direct result, they cannot form secondary tissues. Therefore, they do not undergo secondary growth. Their diameter is pretty much fixed after primary growth finishes, where they're commonly found. They are a characteristic feature of monocotyledonous plants. You can think of them as closed roads, no further widening, no increase in girth once these bundles are formed. This explains why monocots like grasses or bamboo typically don't develop thick, woody stems like you see in dicot trees. It's just a different growth strategy. Okay, beyond just having cambium or not, the arrangement of vascular bundles within the plant body also varies quite significantly, and these arrangements provide key identification features. We mainly recognize two types of arrangement. First, radial arrangement. In this setup, the xylem and phloem are arranged in an alternate manner. They lie along different radii within the central part of the organ. A classic, very clear example of this radial arrangement is seen in roots. Both dicot and monocot roots have this. This design is highly efficient for absorption, ensuring that water and minerals, once taken up, can effectively and quickly reach the central vascular tissue to be transported upwards. Second, we have conjoint arrangement. Here, the xylem and phloem are situated jointly, side by side, along the same radius of the vascular bundle. This arrangement is the most common type you'll find in stems and leaves. Within most conjoint vascular bundles, you'll typically observe that the phloem is located only on the outer side of the xylem, towards the periphery. These distinctions in arrangement, radial versus conjoint, are really crucial for correctly identifying plant organs when you look at microscopic sections, especially in your practical exams. Right, now that we have a pretty solid understanding of the three fundamental tissue systems, epidermal, ground, and vascular, let's try to bring all this knowledge together. We're going to compare and contrast how these systems are organized differently in dicotyledonous and monocotyledonous plants. Specifically, we'll look at their roots, their stems, and their leaves. Pay close attention here, because this comparative study is an extremely high-yield area for competitive exams like NEET. Focus on the specific distinguishing features for each type of plant and organ. This is where you really develop that keen eye for identification that these exams demand. Okay, let's begin with the anatomy of a dicotyledonous root. We'll use the sunflower root as our typical example here. If you were to look at a transverse section, a TS, of a mature dicot root, you'd see a highly organized internal structure. Let's move systematically from the outside inwards. The outermost layer is called the epiblema. It's a single layer of cells. Many of these cells extend outwards to form those delicate unicellular root hairs we talked about. And as we know, these are absolutely vital for maximizing water and mineral absorption from the soil. Moving inwards, just beneath the epiblema, you find the cortex. This region consists of several layers of thin-walled parenchyma cells. A key characteristic here is the presence of noticeable intercellular spaces between these cells. This helps with gas exchange within the cortex and also provides some storage capacity. Now, the innermost layer of the cortex is a very distinctive and functionally critical layer called the endodermis. It's made up of a single layer of barrel-shaped cells. And crucially, these cells are compactly arranged without any intercellular spaces. They form a tight, continuous barrier. What makes the endodermis so special, so important, it's the presence of the Casparian strips. These are unique band-like depositions of a water-impermeable waxy material called suberin. You find these strips on the tangential walls, top and bottom, as well as the radial walls, the sides of the endodermal cells. Now listen carefully, this is a super important feature, a real favorite for exam questions. The Casparian strips effectively block the apoplast pathway. That's the movement of water and solutes around the cells through the cell walls and intercellular spaces. Instead, they force water and dissolved minerals to pass through the endodermal cells themselves via the simplest pathway, meaning through the living cytoplasm. Why is this so significant? Think of it as nature's gatekeeper, a biological checkpoint. It allows the plant to exert absolute control to regulate precisely what enters its vital vascular tissue. It's like a quality check gate, ensuring only necessary substances get through, and it acts as a crucial barrier against potentially harmful things from the soil, like pathogens or toxins. This precise regulation is vital for the plant's health. Immediately next to the endodermis, moving towards the center, you'll find a few layers of thick-walled parenchymatous cells. This layer is known as the pericycle. The significance of the pericycle, immense for plant development. This is the layer from which lateral roots initiate. Branch roots actually grow outwards from here. Furthermore, during secondary growth in dicot roots, when the root gets thicker, the vascular cambium, which drives this thickening, also initiates partly from cells within the pericycle. Okay, right in the very center of the dicot root, the pith is typically small. Sometimes it's even inconspicuous, hard to see clearly. It doesn't form a prominent central core. The parenchymatous cells that are located between the patches of xylem and phloem, those are collectively termed the conjunctive tissue. Now for the vascular bundles themselves. In a typical dicot root, you usually find 2 to 4 xylem patches and 2 to 4 phloem patches. They're arranged in that radiate pattern we discussed earlier, alternating xylem, phloem, phloem. A crucial point specifically for dicot roots is that later on, a ring of cambium develops between these xylem and phloem patches, usually from the conjunctive tissue and parts of the pericycle. This cambium ring is what enables the root to undergo secondary growth, allowing it to increase significantly in diameter and become much more robust over time. Finally, just a collective term you need to remember, all the tissues located on the inner side of the endodermis. This includes the pericycle, the vascular bundles, xylem and phloem, and the pith, however small it might be. All of that together constitutes what is known as the stele.
Okay, so just to confirm my understanding, the stele is essentially the entire central vascular cylinder of the root, containing all the main transport and developmental tissues inside the endodermis.
Exactly. You've got that perfectly. It's the core functional unit, responsible for conduction, and as we just discussed, it's also the origin point for crucial structures like lateral roots. Visualizing the stele helps you understand that central organization of the root structure. Right, now let's pivot, and let's turn our attention to the monocotyledonous root. You'll notice that the anatomy of a monocot root shares quite a few similarities with the dicot root in its basic layout. Yes, it also has an epidermis, a cortex, an endodermis, a pericycle, vascular bundles, and a pith. The basic layers are there. However, there are some very distinct and critical differences, and these are essential for quick identification in your exams. So focus here. First, let's look at the xylem bundles. Remember, the dicot root usually has 2 to 4. Well, in contrast, monocot roots are characterized by having more than 6 xylem bundles, often many more. This arrangement, where there are numerous xylem bundles, is specifically termed polyarch. Poly meaning many, arch referring to the xylem bundles. This is a clear diagnostic feature. Polyarch monocot root. Second, the pith. Remember, the dicot root has a small or often inconspicuous pith. Well, in monocot roots, the pith is notably large and well developed. It provides a prominent central core. This is usually a very clear distinguishing feature you can spot easily under the microscope. Large pith equals likely monocot root. And third, a major functional difference. Monocotyledonous roots do not undergo any secondary growth. This means they don't increase in girth over time, unlike dicot roots, which can become quite thick. Why? Primarily because their vascular bundles are closed, they lack the cambium needed for secondary growth. This distinction is fundamental to understanding their growth pattern. Here's a quick mnemonic that many students find helpful for quick identification during practicals or MCQs. For monocots, think M for many xylem bundles, polyarch, and M for massive pith. This simple trick can often help you differentiate them from dicots almost instantly. Many and massive monocot root. All right, let's move upwards now, away from the root system, and let's thoroughly examine the anatomy of a dicotyledonous stem. We'll focus on a young one, as it shows the primary structure most clearly. If you take a transverse section of a typical young dicot stem, you'll observe a very characteristic and ordered internal structure, quite different from the root. Starting from the outside again, we have the epidermis, the primary protective layer. It's usually covered with a thin cuticle similar to leaves, helping prevent water loss. This epidermal layer might also bear trichomes, stem hairs, and occasionally a few stomata, though usually fewer than on leaves. Moving inwards, beneath the epidermis and stretching towards the vascular tissue, is the cortex. Now, the cortex in a dicot stem is quite substantial. It consists of multiple layers of cells, and importantly, it's organized into 3 distinct subzones. The outermost subzone is the hypodermis. This layer consists of a few layers of collenchymatous cells right below the epidermis. Crucially, remember collenchyma, it provides mechanical strength and flexible support to the young, still growing stem, allows it to bend without easily breaking. Vital for those tender shoots. Below the hypodermis are the main cortical layers. These are usually made of rounded, thin-walled parenchymatous cells. A key feature here is that they have conspicuous intercellular spaces. This facilitates gas exchange within the cortex and provides areas for storing food and water. The innermost layer of the cortex is, once again, the endodermis. But it looks a bit different here compared to the root. The cells of this endodermal layer in the stem are notably rich in starchy grains. So much so that this layer is also commonly and importantly referred to as the starch sheath. It likely plays some role in regulating movement into the stele, but that prominent starch content is its most distinctive feature in the stem. Okay, next we find the pericycle. This layer is situated on the inner side of the endodermis and typically located immediately above the phloem bundles. In dicot stems, the pericycle often appears in the form of characteristic semilunar patches of sclerenchyma cells, like little half-moons. These sclerenchyma patches provide additional rigid support, forming a kind of protective cap over the vascular bundles. Now, radiating outwards from the central pith and located in between the vascular bundles, you'll find a few layers of radially placed parenchymatous cells. These are known as medullary rays. Their main job is to facilitate the radial transport of water, nutrients, and gases moving sideways between the pith and the cortex. They create vital connections across the stem. Right, now for a definitive hallmark feature of the dicot stem, the vascular bundles themselves. You'll observe a large number of these bundles, and they are arranged in a very characteristic ring formation, like beads on a necklace. This distinct ring arrangement is a defining diagnostic characteristic of a dicot stem, something you should immediately look for in diagrams or slides. Ring dicot stem. Each of these vascular bundles is conjoint, xylem and phloem together on the same radius. Crucially, they are also open, remember. Open means cambium is present between the xylem and phloem. And this cambium enables secondary growth, allowing the stem to increase significantly in girth and become woody over time. Furthermore, they have endarched propoxylum. That means the propoxylum, first formed smaller xylem, is positioned towards the pith, the center, while the metaxylum, later formed larger xylem, is towards the periphery, the outside. Finally, occupying the entire central portion of the stem is the pith. In dicot stems, the pith is typically large and conspicuous. It consists of a large number of rounded parenchymatous cells, also characterized by having large intercellular spaces. This central region often serves mainly for storage of food reserves. Okay, let's now compare and contrast this with the monocotyledonous stem. The monocot stem presents several really distinct anatomical features that stand in sharp contrast to the dicot stem. This actually makes them relatively straightforward to differentiate once you know what to look for. First, let's look at the hypodermis again. Remember, the dicot stem's hypodermis was collenchymatous, for flexible support. Well, in monocot stems, the hypodermis is composed of sclerenchymatous cells. Sclerenchyma provides a more rigid, less flexible type of mechanical support. This is crucial for many monocots, which are often tall and slender like grasses or corn, needing to withstand wind without having much secondary thickening for support. Next, and perhaps the most striking difference you'll notice immediately, the arrangement of the vascular bundles. Instead of being neatly arranged in that characteristic ring, a large number of vascular bundles are found scattered throughout the ground tissue, just dotted around seemingly randomly. This scattered arrangement is an unmistakable diagnostic feature of monocot stems. Scattered bundles equals monocot stem. Additionally, each of these scattered vascular bundles is typically surrounded by a prominent sclerenchymatous bundle sheath. This provides extra protection and mechanical support to the vascular tissues within the bundle. Now, the ground tissue itself. The entire interior of the monocot stem, apart from these scattered vascular bundles, consists of a large, conspicuous parenchymatous ground tissue. There's no distinct anatomical separation into a separate cortex, endodermis, pericycle, and pith like you clearly see in dicot stems. It's more of a continuous, undifferentiated tissue filling the space. The vascular bundles themselves, they are conjoint, similar to dicots in that xylem and phloem are on the same radius. However, here's a crucial difference related to growth. They're closed. Closed means no cambium is present within them, and as a direct consequence, monocot stems do not undergo any secondary growth. They don't get thicker over time. This is why monocots like grasses or palms generally don't develop thick, woody stems. They maintain a more or less uniform diameter after their primary growth phase. You'll also often notice a size variation in these scattered bundles. The peripheral vascular bundles, the ones closer to the outside edge of the stem, are generally smaller than the ones located more centrally. A subtle but important difference to note for your exams. Phloem parenchyma is notably absent in monocot stems. Dicot stems usually have it. And finally, a curious feature. You'll often find distinct water-containing cavities present within the vascular bundles themselves, often formed by the breakdown of some protoxylum elements.
Oh, that's interesting. What's the specific purpose of these water-containing cavities in monocot stems? Are they just for storage, or do they have a more complex role?
That's an excellent and insightful question. They're definitely not just empty spaces, no. These water-containing cavities actually serve a vital role, especially for monocots. Many of them, like grasses or tall, fast-growing plants, might face fluctuating water availability, right? So they primarily act as little water reservoirs. They help the plant manage its water supply and maintain turgidity, especially during periods of water stress or scarcity. But beyond just simple water storage, they can also contribute to the overall flexibility and perhaps even buoyancy of the stem. This could be particularly beneficial for tall, slender monocots like bamboo or corn, allowing them to sway in the wind without snapping easily. So yes, they're quite a clever structural adaptation. They provide both hydraulic stability and a degree of mechanical resilience. Right, now let's shift our focus to the leaves. We'll start with the dorsiventral, or dicotyledonous leaf. Dorsiventral means it has distinct upper and lower surfaces. When you examine a vertical section taken through the lamina, that's the flattened blebe part of the leaf, you'll clearly observe three main anatomical parts: the epidermis, the mesophyll, and the vascular system, the veins. Okay, the epidermis. It forms the outermost protective covering, as usual, and it's present on both the upper surface, which we call the adaxial epidermis, and the lower surface, the abaxial epidermis. It has a conspicuous cuticle covering it, that waxy layer that's crucial for minimizing water loss through transpiration. Now, regarding stomata distribution, this is a very important identifying feature for dorsiventral leaves. The abaxial epidermis, the lower surface, generally bears significantly more stomata than the adaxial epidermis, the upper surface. In fact, the adaxial upper surface in many dicots may even completely lack stomata altogether.
Oh, yeah, that's interesting. Is there a specific physiological or maybe adaptive reason why dicot leaves usually have so many more stomata on their lower surface that feels like it might limit gas exchange a bit?
That's a very perceptive question, and it points directly to a brilliant adaptive strategy. It's really a classic example of structure serving function to minimize water loss, which, remember, is a plant's constant battle. Think about it. The lower surface of the leaf is typically less exposed to direct sunlight, intense heat, and strong winds compared to the upper surface, right? So by concentrating most of the stomata on the underside, the rate of transpiration, that loss of water vapor, is significantly reduced. This strategic placement helps the plant conserve water much more effectively, especially in environments where water might be scarce. It's a smart, built-in water-saving mechanism. It prioritizes water conservation without completely shutting down the essential gas exchange needed for photosynthesis. Clever balance. Next, the mesophyll. This is the vital tissue located between the upper and lower epidermis. It forms the bulk of the leaf's interior, and as you'd absolutely expect, it's packed with chloroplasts because this is the primary site where photosynthesis occurs. Now, the mesophyll in a dorsiventral leaf is a key diagnostic feature because it's clearly differentiated into two distinct types of parenchyma cells. First, we have the palisade parenchyma. This layer is adaxially placed, meaning it's positioned towards the upper surface of the leaf, right beneath the adaxial epidermis. It's made up of elongated, often cylindrical cells. They're arranged vertically, packed very tightly, sort of parallel to each other like columns. This layer is exceptionally dense with chloroplasts. Think of these cells as the leaf's primary solar panels. They're optimally oriented and densely packed to efficiently capture the maximum amount of sunlight hitting the leaf surface. Their columnar shape helps light penetrate deeper too. Right below the palisade cells, extending down to the lower epidermis, you'll find the spongy parenchyma. These cells are typically more oval or rounded in shape, and their key characteristic is that they are loosely arranged. There are numerous large spaces and extensive air cavities between them. These air cavities are incredibly crucial for efficient gaseous exchange. They form an interconnected system, like a sponge, allowing carbon dioxide to diffuse rapidly from the stomata, mostly on the lower surface, up to every photosynthetic cell in the palisade layer. And similarly, they allow the oxygen produced during photosynthesis to diffuse out easily towards the stomata. It's like an internal ventilation and delivery system, ensuring gases can move freely within the leaf where needed. Finally, the vascular system of the leaf. This includes the vascular bundles, which are clearly visible to us as the veins and the prominent midrib running down the leaf. The size of these vascular bundles directly relates to the size of the veins they are part of. Larger veins contain larger bundles. Smaller veins have smaller ones. A key reflection of the characteristic reticulate or net-like venation pattern of dicot leaves is that these veins show varying thicknesses throughout the leaf, creating that intricate network. The vascular bundles themselves are typically surrounded by a layer of thick-walled cells called bundle sheath cells. These provide both protection to the xylem and phloem and also some structural support to the vein. Okay, now let's move on to the isobilateral or monocotyledonous leaf. Isobilateral means both surfaces are more or less similar structurally and functionally. Think of a grass blade. The anatomy of an isobilateral leaf shares many fundamental similarities with the dorsiventral leaf. Yes, it possesses an epidermis, mesophyll, and a vascular system. However, it exhibits several characteristic differences that are exceedingly important for differentiation in your exams. First, let's look at stomata distribution again. In an isobilateral leaf, stomata are typically present on both the adaxial upper and abaxial lower surfaces of the epidermis, often in roughly equal numbers. This bilateral distribution is quite different from the predominantly lower surface stomata we usually see in dicot leaves. Second, mesophyll differentiation. This is a very key distinguishing feature. The mesophyll in a typical monocot leaf is not differentiated into those distinct palisade and spongy parenchyma layers that we saw so clearly in dorsiventral leaves. Instead, it's generally a more uniform tissue throughout, composed of parenchyma cells containing chloroplasts, but without that clear layering. And third, a very specific and fascinating adaptation found predominantly in grasses, which are, of course, prime examples of monocots: the presence of bulliform cells. In many grasses, certain adaxial upper surface epidermal cells, particularly those located in groups along the veins, modify themselves. They become large, empty-looking, and colorless cells. These are precisely what we call bulliform cells. Let's understand the brilliant function of these bulliform cells step by step, because it's a really powerful example of adaptation to the environment, especially water availability. Okay, when these bulliform cells have absorbed sufficient water and are turgid, meaning they are full of water and swollen, pushing outwards, they cause the leaf surface to be fully exposed, flattened out. This maximizes the surface area available for light absorption and photosynthesis, good conditions. However, when the plant is under water stress, or there's a scarcity of water, these specialized bulliform cells rapidly lose water. They become flaccid or limp. Now, when they become flaccid because of their position and structure, they cause the leaf blade to curl or roll inwards, usually along its longitudinal axis, like rolling up a piece of paper. The significance of this curling mechanism, it's immense. It's a sophisticated, built-in drought response system. By curling inwards, the leaf dramatically minimizes its exposed surface area to the sun and drying wind. This significantly reduces the rate of water loss due to transpiration during dry conditions. Imagine them as tiny, natural umbrellas that automatically close up when the weather gets too dry, protecting the more sensitive inner parts of the leaf from excessive water loss. It's a very clever and highly effective adaptation for survival in environments where water might be scarce periodically, like grasslands. Lastly, the characteristic parallel venation of most monocot leaves is clearly reflected in their internal anatomy as well. When you observe a vertical section, you'll generally see that the vascular bundles, the veins, are of nearly similar sizes throughout the leaf section. This reflects the uniform nature of parallel veins, unlike the varying thicknesses of veins you see corresponding to the reticulate venation pattern in dicot leaves. All right, so to quickly summarize our comprehensive deep dive into the anatomy of flowering plants today, we've meticulously explored how these complex organisms are organized, right from the basic cellular unit up to specialized tissues, and then into distinct organs like roots, stems, and leaves. We delved into the three fundamental tissue systems: the epidermal system, acting as that clever outer protective shield; the ground tissue system, forming the main bulk and doing vital jobs like storage and photosynthesis; and the vascular tissue system, the plant's remarkably efficient transport network for water, minerals, and food. And most importantly, we've dissected in great detail the specific anatomical differences between monocot and dicot roots, stems, and leaves. Remember, these structural differences, they aren't just random. They are usually ingenious adaptations to their environments and their diverse ways of life. They are absolutely crucial for their survival and success. Now, to truly master this chapter for your upcoming board exams, and especially for NEET, make sure you revise these distinguishing features repeatedly, again and again. A highly effective strategy, which I strongly recommend you do, is to create detailed comparison tables. Compare monocots and dicots side by side for each organ, roots, stems, and leaves. This side-by-side comparison will make differentiation much easier when you're under exam pressure. It helps you quickly recall those critical points. Also, please practice drawing and labeling the transverse sections of dicot and monocot roots, stems, and leaves from memory. Don't just look at the book. An accurate, well-labeled diagram. That can fetch you full marks on a question, and honestly, it really solidifies your understanding like nothing else. And finally, don't forget to practice past year questions. Specifically, look for questions based on frequently asked topics like the stomatal apparatus, those unique Casparian strips in the root endodermis, and the various types and arrangements of vascular bundles: open versus closed, radial versus conjoint, ring versus scattered. These are consistently high-scoring areas, Keep revisiting these concepts. Maintain your consistency in your efforts. Remember, consistency is absolutely key to cracking competitive exams like NEET. Please do reach out if you have any further doubts or questions on this chapter as you revise. Keep learning, keep pushing forward, and all the very best for your preparations.